If you were to count every single bacteriophage on Earth, you'd reach a number that breaks human comprehension: 10 nonillion. That is a 1 followed by 31 zeros. This chart shows the estimated global populations of different life forms. As you can see, when you stack that 10 nonillion against every other living organism on the planet, including bacteria, animals, and humans combined, the phages completely dwarf them. A bacteriophage, or phage for short, is a highly specialized virus. But unlike the viruses that give us colds or the flu, phages have evolved exclusively to infect and replicate inside bacteria. Because their targeting systems are so specific to bacterial cells, they are completely harmless to humans. You have billions of them inside your body right now. To our oldest bacterial pathogens, however, they are the ultimate natural assassin. This diagram illustrates the first step of a process called the lytic cycle. A phage floats through the environment until it randomly bumps into a bacterium and locks its tail fibers onto specific, matching receptors on the cell wall. Once firmly attached, the virus acts like a microscopic hypodermic needle. It punctures the bacterial membrane and forces its own genetic materials straight inside the host. Inside, the bacterial ribosomes, the cellular machines that usually build proteins for the bacterium, are forced to stop their normal work. Instead, they read the viral DNA and begin rapidly manufacturing new phage parts. Those newly synthesized heads, tails, and fibers don't stay in pieces for long. They spontaneously snap together, constructing dozens to hundreds of fully functional virions right there in the cytoplasm. The physical pressure and viral enzymes finally cause the bacterial cell wall to rupture, or lyse. The host cell dies, and a massive swarm of new phages spills out into the environment. One virus enters, and hundreds leave, and the host is destroyed. This self-replicating cycle makes phages highly efficient, precision-guided weapons against bacterial infections. Recognizing this destructive precision, early 20th century scientists developed phage therapy, the medical practice of deliberately applying these specialized viruses to clear out pathogenic bacteria in humans. Researchers in the Soviet Union and Eastern Europe pioneered this field. They successfully treated soldiers and civilians for bacterial diseases like dysentery by cultivating specific phages in the laboratory. But the outbreak of World War II demanded medical treatments that were instantly deployable to the front lines. Around the same time, Western scientists discovered and purified the chemical antibiotic penicillin. Antibiotics held a massive logistical advantage. Chemical compounds are easy to mass-produce, package, and ship. Phages, on the other hand, were living entities that required careful cultivation and exact matching to specific bacterial strains. This ease of industrial production sparked a boom. Western medicine filled pharmacy shelves with these broad-spectrum drugs and almost entirely halted its research into viral therapies. Grave. By prioritizing the convenience of a chemical pill that kills indiscriminately over the precision of a targeted virus, the global medical system created a critical vulnerability. Bacteria reproduce quickly and mutate often. Over decades of heavy global antibiotic use, they have evolved biological defenses to survive our chemical drugs, a phenomenon known as antibiotic resistance. The scale of this problem is severe. The Centers for Disease Control estimates that nearly 3 million of these antibiotic-resistant infections occur every year in the United States alone. This chart visualizes the projected death toll from antibiotic-resistant superbugs. As our standard chemical treatments continue to fail, the annual number of fatalities is climbing rapidly. Because our miracle drugs are losing their effectiveness, the medical community is now forced to look back a century and re-examine the natural bacterial predators they previously left behind. That re-examination is already saving lives. In 2016, a patient named Tom Patterson contracted a deadly, multi-drug-resistant bacterial superbug. Every conventional antibiotic failed to stop the infection. He fell into a coma and was nearing death when his medical team at UC San Diego decided to try an experimental intervention. They rapidly isolated the specific strain of bacteria attacking Patterson's body and matched it with a tailored intravenous cocktail of bacteriophages designed specifically to hunt that exact microbe. The viral cocktail worked. The phages tracked down and eliminated the superbug, leaving his healthy human cells and beneficial gut bacteria completely unharmed. Within days, Patterson woke from his coma. By harnessing the highly specific biological rules of the lytic cycle, physicians cured an infection that modern chemicals could not touch. To survive the resistant superbugs of tomorrow, medicine must learn to partner with nature's oldest predators.